Bismillahirrahmanirrahim. The topic of today's lecture is internal structure of the Rhinencephalon, part of the brain which is concerned with the sense of smell or olfaction. There are many odorous molecules in the air which enter the nasal cavity when we inhale the air, and the olfactory mucosa is in the nasal cavity, and these neurons of the olfactory mucosa, they are stimulated and the impulses pass to the olfactory cortical areas, and we feel smell sensation. Now the olfactory system, it comprises of different parts. Many parts we discussed in the external features also, so we will again repeat that. So olfactory system consists of olfactory nasal mucosa, then the first cranial nerve, olfactory nerve, then different parts of the brain collectively known as Rhinencephalon. Because Rhinencephalon is concerned with the sense of smell, so Rhinencephalon is also known as smell brain. There are different components of the Rhinencephalon or smell brain. These are olfactory bulb, olfactory tract, olfactory tract divided into three roots, also known as tree. Then pre-piriform area, the pre-piriform area further comprise of the pre-piriform area, and next to pre-piriform area is piriform area. And the pre-piriform area further comprise of different parts like anterior perforated substance, anterior part of para-hippocampal gyrus, next is uncus, then amygdaloid body, these are different parts of the piriform area. Next is hippocampal formation. which further comprise of pes hippocampus, or simply hippocampus, then fiber bundles like alveus, fimbriae, crura of the pharynx, body of the pharynx, and column of the pharynx. Column of the pharynx terminate into the mammillary bodies. And from mammillary bodies, mammillothalamic tracts arise, which are in the thalamus, the anterior and dorsomedial nuclei of the thalamus. Next component of the piriform area. Is paraterminal gyrus. And habenular complex. These are different parts of the smell brain or rhinencephalon. Now briefly we'll discuss all these parts one by one, starting with the olfactory mucosa. Now nasal cavity. Longitudinal section through the head. Here is between nasal cavity and oral cavity is the palate. This is hard palate and soft palate. Neck region. Below the palate, this is oral cavity and its contents: tongue, teeth, etc. Above is the nasal cavity. Here the roof and floor of the nasal cavity is shown. Now the nasal cavity mucosa we divide into various regions. Here this part of the nose which is dilated, this is vestibular area. Vestibular region of the nasal cavity. Then the greater part of the nasal cavity, this is the respiratory region. Of nasal cavity. And the upper smaller part of the nasal mucosa, which line the roof of the nasal cavity and upper parts of the lateral and medial walls of the nasal cavity. This is olfactory mucosa, olfactory region. Now this vestibular region, the inner surface of the lower dilated part, vestibules, is lined by mucosa, which is epidermis, that is stratified squamous keratinizing type of epithelium with the sweat glands, sweat, sebaceous glands, and hair follicles. Then the respiratory region, this is lined by pseudo-stratified ciliated columnar epithelium. Pseudo-stratified ciliated columnar epithelium and goblet cells, mucus secreting goblet cells. The respiratory region. And the upper smaller part is the olfactory region, lined by olfactory mucosa. It is also pseudo-stratified epithelium. Now this olfactory mucosa, line the roof of the nasal cavity and the upper parts of the lateral and medial walls. And this bony plate belongs to the skull, which show perforations. So this is cribriform plate. Of ethmoid bone of the skull. And this is cranial cavity, which is occupied by brain. And here on the basal surface of the brain is the olfactory bulb, followed by olfactory tract. It's attached to the lower surface of the brain. And this is the orbital surface of the cerebral hemisphere. On the orbital surface of the frontal lobe is the olfactory bulb and olfactory tract. below the cribriform plate of ethmoid bone, which show perforations for the passage of filaments of the olfactory nerve, is the olfactory mucosa. Now, olfactory mucosa. On the nasal surface, there is a connective tissue layer called lamina propria. Connective tissue, lamina propria, comprised of connective tissue fibers, connective tissue cells. There are blood vessels. And there are mucus secreting glands, mucus glands. And above this is the olfactory mucosa. It comprises of three types of cells: the olfactory neurons, the sustentacular cells, and the stem cells. Suppose these are olfactory neurons. These olfactory neurons are supported by sustentacular cells. Sustentacular cell, and in the basal region there are basal cells. This is the structure of olfactory mucosa. Now, olfactory neurons, detailed structure. These neurons are bipolar neurons. This is the cell body of an olfactory neuron. From it, processes arise called neurites. This is an axon, and this is dendrite. Cell body of an olfactory neuron. This is axon, and this is dendrite. So the olfactory neurons are bipolar neurons. The point of the cell body from which a process arises is known as pole. So because here only two processes arise, a dendrite and axon, so and the body of the olfactory neuron has two poles. This is why they are called bipolar neurons. Dendrites arise from the basal part of the body. In the lower portion, the dendrite expands to form olfactory vesicle. Then from the olfactory vesicle, cilia arise, fine cytoplasmic processes. Cilia arises, and these cilia, they run parallel to the lower surface of the of olfactory mucosa. You can see they run in this direction, not vertically. In the connexu lamina propria, these are cilia of these olfactory neurons, which run parallel to the surface, not vertically at right angle. Now the cell membrane of the olfactory cilia. Number one, these cilia do not have, these cilia are not motile. Non-motile cytoplasmic processes, although they have microtubules. The cilia are motile because of presence of microtubules, but the special feature of these cilia of the olfactory neurons, they have microtubules, but they are non-motile. Their cell membrane, because cilia are cytoplasmic processes, cell membrane inside cytoplasm flows, and still inside the cytoplasm are the microtubules, etc. So the cell membrane of cilia of olfactory neurons have odorant binding proteins. So embedded in the cell membrane of cilia there are odorant binding proteins. So they act like olfactory receptors, these cilia of the olfactory neurons. Now, olfactory neurons, as I told you, are bipolar neurons. They have some special features. Of olfactory neurons. Number one, they have non-motile cilia, and the cell membrane of cilia have odorant binding proteins. Therefore, they act as olfactory receptors. Number three, the olfactory neurons can divide by mitosis. They replicate by cell division because they have centrioles. The centrioles of the cells, they help in cell division, forming a mitotic spindle. So the cells replicate. Now the immature neurons, they have centrioles, therefore the immature neuron can replicate. Why? Because they have centrioles. The mature neurons, they do not have centrioles. So once a neuron become mature, their centrioles disappear, then they cannot replicate, they cannot undergo mitosis. With one exception, it is only the mature olfactory neurons which can divide. The olfactory neurons are mature neurons, and these are the only neurons which can replicate because they have the centrioles inside. The lifespan is not more than, the lifespan of the olfactory neurons is not more than 2 months. The lifespan of olfactory neuron is not more than 2 months. They die, and they must be replaced. They are replaced by these basal cells, which are also believed to be the stem cells. And second, when the olfactory neurons they are damaged, they can replicate by division because they have centrioles inside. So olfactory neurons are the only mature neuron in the body which possess centrioles, and they can divide. These were some special features of the olfactory neurons. The axons of these olfactory neurons, they pass through the cribriform plate of ethmoid bone. Suppose this is olfactory neuron, one olfactory neuron in the olfactory mucosa. And another olfactory neuron embedded in the olfactory mucosa. The dendrites and the axons. These axons pass through the openings in the cribriform plate of the ethmoid bone, enter the cranial cavity, and enter the olfactory bulb. These filaments of the nerves, they form the olfactory nerve, the first cranial nerve. Then in the olfactory bulb, there are also neurons in the olfactory bulb. So the olfactory neurons make synapses with the neurons in the olfactory bulb, with the dendrites of the neurons in the olfactory bulb, and from it the axons arise, which are crowded together and run as olfactory tract. Continue into the different parts of the Rhinencephalon. So the olfactory nerves are axons of the olfactory neurons, which are embedded in the nasal olfactory mucosa. These axons enter the cranial cavity through cribriform plate and enter the olfactory bulb, where they make synapses with another set of neurons, with the dendrites of these neurons in the olfactory bulb. Then there are the axons of these neurons then pass towards the other parts of the rhinencephalon, forming a tract known as the olfactory tract. These olfactory neurons in the olfactory mucosa are also known as the primary olfactory neurons. The olfactory neurons in the olfactory mucosa are also known as primary olfactory neurons. Then these olfactory neurons, which start in the olfactory bulb, these are known as secondary olfactory neurons. And the axons of the secondary olfactory neurons form olfactory tract, then different striae, and then enter different parts of the rhinencephalon. Next to the olfactory neurons in the olfactory mucosa are sustentacular cells. These are supporting cells. They are meant for physical and nutritional support to the olfactory neurons. And in the basal region, I told you there are basal cells which act like stem cells. And then there is connextive lamina propria, in which there are different structures, including the mucous glands. The Sustentacular cells are considered as glial cells. So you can see the nervous tissue, all components are here. The olfactory neurons in the olfactory mucosa, and then the glial cells known as the sustentacular cells in the olfactory mucosa. Now, another very important property as characteristic feature of the this olfactory neuron is that these olfactory neurons is that these are the only neurons olfactory neurons are the only neurons which have direct contact with the external environment. So the olfactory neurons in the nasal cavity, they are exposed to the outside environment directly. These are the only neurons in the human body. So because these neurons, they occupy the olfactory nasal mucosa, and which have olfactory neurons and the glial cells in the form of sustentacular cell. This is why olfactory mucosa is also known as nose brain. Nose brain. Then in the lamina propria there are mucus secreting glands, and this is why that the nasal surface of olfactory mucosa, and of course rest of the mucosa also, is lubricated by thin film of mucus fluid. Thin film of mucus fluid. Now the odorant molecules these are released into the air in the form of molecules or particles. Odorant molecules emitted from any object. They enter the air when the air is inhaled, enter into the nasal cavity. Now these odorant molecules, when they are dry, they are unable to stimulate the olfactory neurons. So this is why these molecules, when they enter the nasal cavity, they are dissolved in the mucus lining the olfactory mucosa, and these odorant molecules in fluid form are then able to stimulate the olfactory receptors, that is the cilia of the neurons, and then the olfactory neurons are stimulated and impulses go to the centers. So this is the importance of these mucous glands embedded in the lamina propria of the olfactory mucosa. Now sensations, you know, we have general sensations and spatial sensations. General sensations, touch, pain, temperature, etc. And spatial sensation, smell, taste, etc. Now there is a pathway. For example, this is the cerebral cortex, sensory cortex. Midbrain, pons, medulla oblongata, and spinal cord. Cerebellum. So if we cut a section of the spinal cord, the gray matter inside anterior horn and posterior horn. Then we have spinal nerve, dorsal root, characterized by presence of sensory ganglion or spinal ganglion, and ventral root, joined together to form spinal nerve. So various sensations, they are travel through sensory fibers to the brain and spinal cord. Now in the sensory ganglion there are unipolar neurons, sensory neurons, which has a peripheral process and central process. Central process enter the spinal cord and brainstem. Spinal cord in case of spinal nerve and brainstem in case of sensory components of the cranial nerves. So various sensations from the different parts of the body reaches the posterior horn of the spinal cord, which is sensory in function. Anterior horn is motor. Then from here another neuron arises from the posterior horn, which ascend through the brainstem. And relay in the thalamus. From the thalamus, another neuron starts, which relay in the sensory cortex. So in this way, from periphery, the sensations are carried to the sensory cortex. Now, from periphery to the sensory cortex, one can see that there are 3 neurons involved. This is first order neuron. The cell body of this neuron is the spinal ganglion, and peripheral passes carrying sensation from the periphery, and the dendrite of this neuron of the spinal ganglia relay in the posterior horn. From the posterior horn, another neuron you can see it starts and carry these sensations toward the thalamus. This is second order neuron in the sensory pathway, and this neuron relay in the thalamus. From the thalamus, a third order neuron starts, which relay in the sensory cortex. So in all sensory pathways, touch, pain, temperature, etc., 3 are involved from periphery towards the sensory cortex. First order neuron, second order neuron, third order neuron. But the olfactory sensation, which is a spatial sensation, has this property that only 2 neurons are involved here. You can see here olfactory neuron from the olfactory mucosa relay in the olfactory bulb. This is first order neuron in the olfactory pathway. First order, and the first order neuron dendrite relay in the olfactory bulb, and from the olfactory bulb another neuron starts. This is second order neuron. And the axons of the second order neuron terminate in the olfactory cortex. So the olfactory pathway differ from rest of the sensory pathway, that instead of 3 order neurons, it comprises of only 2 order neurons. And other important difference, I told you, all the sensations before reaching the sensory cortex are relayed in the thalamus, except olfactory pathway, the olfactory nerves carrying olfactory pathway do not relay in the thalamus. They pass to the olfactory cortex directly. So this was the olfactory mucosa. And then now the next part is the olfactory bulb. So we discussed the olfactory mucosa, then olfactory bulb, and now olfactory bulb. The olfactory bulb, part of the Rhinencephalon. Olfactory bulb, it is on the basal surface, orbital part of the basal surface of the cerebral hemisphere.
olfactory bulb, followed by olfactory tract, which divide into stria or roots. Olfactory bulb,
and the orbital part of the inferior surface of the cerebral hemisphere, in which the axons of the olfactory neurons or olfactory nerve relay, and from which the second order neuron or secondary olfactory neuron starts. And the axons of the secondary olfactory neuron, they are crowded together to form a tract known as the olfactory tract. The olfactory tract, the distal end, divide into three roots, striae. Three roots are three striae. This is lateral stria or root. This is medial stria. Of the olfactory tract, and this is intermediate stria or intermediate root of the olfactory tract. The secondary olfactory neurons, they start in the olfactory bulb, and their axons traverse distally as the olfactory tract, and the fibers of olfactory tract then divide into lateral, intermediate, and medial striae or the roots.
Now, if we draw an enlarged view of the olfactory bulb, olfactory tract, and these three roots are striae.
This is olfactory bulb of one side.
And the olfactory bulb of the opposite side, dividing into medial stria, intermediate, and lateral stria.
This distal part of the
olfactory tract is enlarged to form olfactory trigone. Olfactory bulb, olfactory tract, olfactory trigone, is divided into lateral stria or lateral root, then intermediate stria, and then medial stria. This is suppose axon of the primary olfactory neuron, olfactory nerve. Then this is secondary olfactory neuron with cell bodies in the olfactory bulb, and their axons, they traverse the olfactory tract. These are secondary olfactory neurons, and these are olfactory nerves, which are axons of the primary olfactory neurons. And these are the cell bodies which are in the olfactory bulb, cell bodies of the secondary olfactory neurons, and these are the axons which traverse to the olfactory tract. These then divide into these three roots: lateral root, intermediate root or stria, and the medial root or stria. This is lateral stria, and this is medial stria, and this is intermediate stria. Same is here. There are several such neurons. Now, fibers of the lateral stria, they relay in the uncus. Fibers of the intermediate stria, they relay in the anterior perforated substance. And the fibers of the medial stria, they relay in the paraterminal gyrus. Same is the case here. Uncus, anterior perforated substance, and paraterminal gyrus. Now, some of the fibers of medial stria, they passing through the medial stria, they pass through the anterior commissure. Suppose this bundle of commissural fibers, anterior commissure. These fibers of the medial stria, some of the fibers,
They enter the anterior commissure and then enter the medial stria of the opposite tract and make synapses with the neurons of the opposite olfactory bulb. And similarly here, in this way the olfactory bulbs are interconnected by these commissural fibers. These are few fibers of the medial stria which pass through the anterior commissure and enter the opposite olfactory bulb. Now this was detail about the olfactory tract and olfactory stria. Now pre-piriform area. What is pre-piriform area? So here, just inner to the medial stria on the orbital surface of the frontal lobe, a small area of the cortex known as the pre-piriform area. The cortex just medial to the medial stria on the basal surface of the cerebrum, frontal lobe. This is known as the pre-piriform area. So this was the pre-piriform area, which is a small part of the cortex just medial to the medial stria on the orbital surface of each cerebral hemisphere. Next, the piriform area comprises of anterior perforated substance. Now, what is the anterior perforated substance? Just distal to the stria, the gray matter show perforations on each side, on the orbital part of the inferior surface of each cerebral hemisphere. This is anterior perforated substance. Now the anterior perforated substance is a thin layer of gray matter which show perforations. Through it, the central branches of anterior and middle cerebral arteries enter. To supply the deeply seated nuclei in each cerebral hemisphere. So the anterior perforated substance is a perforated area of gray matter. It shows perforation for the entrance of central branches of anterior middle cerebral artery which enter each cerebral hemisphere to form the nuclei. And second important point about the anterior perforated substance is the feature that some of the major nuclei of the basal ganglia are continuous with the anterior perforated substance. They project from anterior perforated substance into the white matter of each cerebral hemisphere. This we have discussed with basal ganglia in detail in last lecture. So it is also part of the Rhinencephalon, anterior perforated substance. Next, anterior part of the parahippocampal gyrus and uncus. Now the medial surface of the cerebral hemisphere this is a section through corpus callosum.
And there are different midline structures. Here is the cingulate sulcus and cingulate gyrus, supraspinatus sulcus, parieto-occipital sulcus, collateral sulcus, and then rhinal sulcus. Now, here you know is the cingulate gyrus. Then isthmus, and next to it is parahippocampal gyrus. Which is continued as uncus, hook-shaped gyrus, uncus. So this anterior part of parahippocampal gyrus visible on the medial surface. It's an important part of rhinencephalon. And then next is the uncus, the anterior continuity of the parahippocampal gyrus is continued as hook-shaped gyrus, surrounded by a hook-shaped sulcus known as the rhinal sulcus. This is uncus, and it forms bulk of the piriform area. It forms bulk of the piriform, uncus, and it is also act as primary olfactory cortex. Primary olfactory cortex. So anterior perforated substance, anterior part of the parahippocampal gyrus, uncus are parts of the piriform area. Next, amygdaloid body, already we discussed with the limbic system, and amygdaloid body. From it, a bundle of nerve fibers arise known as the stria terminalis. Amygdaloid body, stria terminalis. These bundle of fibers run along with the course of caudate nucleus. Now, some of the fibers of stria terminalis, they run backwards and downwards and terminate in the habenular nuclei. These fibers, which leave the stria terminalis and enter the habenular nucleus, these are known as the habenular stria. Stria habenularis. Remaining fibers of the stria terminalis continue and
divide into 3 bundles. In the septal region, these fibers are known as supracommissural fibers. And then anterior commissural fibers. Then infracommissural fibers. The supracommissural fibers connect with the septal nuclei. Anterior commissural fibers, they enter the anterior commissure. While infracommissural fibers, they enter the anterior part of hypothalamus. So this was just a summary of the striatum fibers arising from amygdala body, and these are its destination. So these were all parts of the piriform area, which is a major part of the rhinencephalon. Next, hippocampal formation, and this already we discussed 3 or 4 times in the last lecture. It comprises of various parts. That is pes hippocampus. Right or left, pes hippocampus. From it, bundle of fibers arise, known as the alveus. Alveus continue as the femori. Femori continue as the crura of the fornix. Crura continue as body of the fornix. Body of the fornix continue as column of the fornix. And column of the fornix terminate in the mammillary body. These are some components of the hippocampal formation. Pus hippocampus, alveus, then femoria, then crus of the fornix, body of fornix, column of fornix, then mammillary body, and the 2 crura interconnected by the commissure of the hippocampus. And apart from these components, there are also some other gyri and fiber bundles. Then paraterminal gyrus. Here on the middle surface of cerebellum and sphere, there is a vertical gyrus known as the paraterminal gyrus. This is corpus callosum. Then fornix, body of the fornix, septum pellucidum, anterior commissure, lamina terminalis, optic chiasma, posterior wall of the third ventricle to which the pineal body is attached, posterior commissure, tuber cinereum and pituitary gland, then right and left mammillary bodies. So you are familiar with our structures. Now here this is lamina terminalis. Now just in front and parallel to the lamina terminalis, there is a gyrus. This is paraterminal gyrus. Can be seen on the medial surface of the cerebral hemisphere. So this was about the paraterminal gyrus. Next, habenular complex. This is the posterior commissure and posterior wall of the third ventricle, pineal body, and this is lateral view of the midbrain. There is superior calculus or inferior calculus. So here in the lower part of the posterior wall of the third ventricle, just above the superior calculus, there is a triangular area in which on each side there is habenular nucleus. One right, one left. This is lateral view, so one nucleus is shown. Now it is located in the habenular trigone, a small triangular depression just above the level of superior caliculi of midbrain. So if we draw a posterior view of the midbrain, a pair of superior caliculi, a pair of inferior caliculi with cruciform sulcus, frenulum, right tracheal nerve, left tracheal nerve, and here there is a depression called habenular trigone, and on each side is the habenular nucleus. And these two habenular nuclei are interconnected by a bundle of commissure fibers known as the habenular commissure. So this is a location and a little detail about the habenular complex. So these were different parts of the Rhinencephalon. Now what is the olfactory cortex? Olfactory cortices, we divide them into primary olfactory cortex and secondary olfactory cortex. Now primary olfactory cortex, mainly it is the uncus. And the secondary olfactory cortex is the lateral part of orbital surface of frontal lobe. This is orbital part of the frontal lobe. So here the lateral part of the orbital portion of the frontal lobe, this is secondary olfactory cortex. And here I have shown the uncus, which is the primary olfactory cortex. The uncus form the bulk of the piriform area of the Rhinencephalon. The primary olfactory cortex, it is responsible for conscious awareness of smell. And what is the function of secondary olfactory cortex? It is responsible for identification and discrimination of smell. The primary olfactory cortex just receive the smell and one become conscious of smell. What about the detail of the smell? It is not determined by the primary olfactory cortex. It is the role of secondary olfactory cortex which further identify the smell and discriminate it. Bad smell, good smell, etc. Various, and which type of smell are determined by this. So this was about the olfactory cortices. So you have seen in the whole olfactory pathway or smell pathway, the sensations are not relayed in the thalamus as in case of other sensations. Now what are the applied aspects? The lack of smell sensation is known as anosmia. Now it has different types. Number one, specific anosmia. There is inability to identify a specific smell, not all smell. One of the smell or two of the smell, they are unable to be identified. Rest of the smell can be identified. So this is called specific anosmia. It is because of the odorant proteins embedded in the wall, absence of odorant binding proteins in the wall of cilia. So for a specific smell, so that smell cannot be identified by patient. Such type of anosmia is called specific anosmia. There is defect in the protein binding odor absence in the wall of cilia of olfactory neurons. Second type is the general anosmia. When there is damage to olfactory path, when there is damage to the olfactory pathway, it results in this. So it is usually unilateral because there is more possibility that the olfactory pathway may be damaged on one side and not on the other side. So one of the features of it is that it is unilateral because of damage to the any part of the olfactory pathway. Then there is another type of anosmia, hyposomus. Hyposomus anosmia. In it, this sense of smell is diminished. And it is usually because of infection, nasal infection. For example, common cold. And it is transient. When the infection is treated, then again sense of smell remain intact. So these are different aspects of applied aspects of the olfactory pathway. So with this we finish with the Rhinencephalon. And I told you that the forebrain comprise of the telencephalon, Rhinencephalon and Diencephalon. We finished with the internal features of the telencephalon, then we finished with the internal features of the Rhinencephalon, and next we will switch on to another part of the forebrain, that is Diencephalon, which we'll discuss in the next coming lectures. Thank you very much.